We have this really cool program on campus called the FIRE program. It's first year research experiences. All right, so it's specifically geared for first year students to apply to get some research experience. I did this when I was here. I thoroughly enjoyed it. I would highly recommend if you're interested in research to check this out. So there is an information session happening pretty soon, I believe. Yeah, next Monday at 8 o'clock. So if you're interested in it, this is one of, I think, 3 information sessions. It's a pretty cool program. Check it out. As far as homework goes, all right, we do have another quiz this Friday. It's just going to cover the material that we get through today. So we might not get through the entire PowerPoint today. So wherever we end up, that's the material it'll be covered, as well as the lecture from Monday. And then assignment 4, just a reminder that is due next Monday in class. I want a printed out copy. That way you can show your work on how you arrived at all those answers. Any questions about homework? All right. So we're going to move right into mechanisms for evolution. All right. So on Monday we talked about the Hardy-Weinberg equilibrium. And the important thing with Hardy-Weinberg is that no evolution is occurring if we are at a Hardy-Weinberg equilibrium. All right, so all that math we did was for populations that was not evolving. All right, now we know that to be not very true in nature. All right, very rarely are we going to find a population that's not evolving. So if we look at the requirements for Hardy-Weinberg, we see that there are several different requirements. And we're going to go through most of these today. I'm not going to go through mutations, though, because we have talked about that several times already, so I don't want to just say the same things for the 3rd time. But we're going to start with talking about infinite population size. All right, so if we're in equilibrium, we're assuming that there's an infinite population size and that no genetic drift is happening. All right, so what is genetic drift? So genetic drift is a change in allelic frequency due to random chance. All right, we're not acting upon a phenotype. We're not having influences by predators on a phenotype. It's completely unrelated to fitness. It's totally by chance. All right, so maybe we had a small population of beetles that were orange and green, and somebody walked along and their shoe accidentally stepped on all the green ones. All right, that's a totally chance event that happened. All right, now when we have genetic drift, we typically find that we're going to favor either the loss or the fixation of a specific allele. And we can see that in this graph here. I have this population of mice, all right, and for the population up top here, all right, we have fixed that brown coloring in the mice, and we have lost the white coloring. Right, exactly the opposite for the bottom population. We have fixed the white coloring and lost the brown coloring. Right, so in this population, no more brown individuals can occur unless we're introducing a brown individual. Now this tends to happen really fast in small populations. Right, so if we look at the red lines on this graph, right, this is a population of only 10 mice for either brown or white. And we see that we get there after about 30 generations. We find that all of our mice are white. And at about 50, all of our mice are brown. Right? But if we look at this middle line, where we have a population of 1,000 mice, right, we see that maybe there's a trend that one allele is favored over the other. All right? But it's going to take a long time to get there. All right? So this happens much quicker in smaller populations. In fact, that you can go through. It gives you some background information, but then you can play around with population size. All right, so right now there's a population size of 1,000 after 200 generations, and the allelic frequency for both four alleles is 0.5. If I were to plot that, all right, I would see after 200 generations, maybe I see a trend towards a fixation. All right, not quite there yet. If I were to change this to say only 10 individuals and plot that, we see that very quickly, very quickly, we got fixation of our allele. All right, so I encourage you to play around with that if you want to get your hands-on experience at population sizes. All All right, but when we're talking about genetic drift, there's kind of these two different ways that we can get genetic drift to rapidly alter allele frequencies. All right, one of which is a bottleneck effect, and the other is a founder effect. So maybe you've heard these terms before. We're going to talk about the bottleneck one first. All right, so this is where a population has been dramatically reduced from some sort of event and then rebuilds. All right, and that's just going to randomly eliminate a specific genotype. Right? So we're not acting upon a phenotype. It's still all due to random chance here. So I have this little picture of this pond full of frogs. Right? We have 3 different phenotypes showing through in coloring. Right now, for some reason, we had a massive drought in this pond, and it just so happens, by random chance, that the frogs that were left in the pond that was remaining happened to be this yellow and like tannish, brownish color there. Right? All the green frogs happened to die out. Totally by chance, had nothing to do with the way they were colored. Now, when that population rebuilds, we've lost that green phenotype, so the population has less genetic variation because we have one less phenotype that's expressed through. Right, a very classic example of bottleneck. And I know there's a lot of text on here. You can read this on your own if you'd like. But it's cheetahs. Right? Cheetahs went through a couple of dramatic bottleneck events. One of which happened through migration. They migrated to a different area and started out with another small population. But another one was that they lived through this mass extinction event. Right? And at one point, it's thought that these cheetahs we only had 7 cheetahs left in all of Africa. Right? So we greatly reduced our population. But over time, these cheetahs have rebounded and built their population back up to what they are today, which I believe in here says it's about 8,000 individuals, which still isn't great, but compared to the 7 we started with is awesome. All right? So they went through a major bottleneck event here. All right, so the other event is a founder event, all right, or the founder effect. And this is where we have a population of individuals, and a subset of those individuals is going to move to another location and start a new population. All right, so I have this picture of the butterfly population up here. It just so happened that some of these butterflies decided to go to a different island and start a new population. Right? It just so happened that they were orange and black. None of the white individuals moved for whatever reason. Totally random. Right? So this new population is only going to have orange and black individuals. We have lost that white phenotype. All right, so when we have this founder's effect, all right, the population that's resulting from the original population tends to have a little less genetic variation than the original. All right, so notice we lost the white phenotype here. Maybe only black butterflies moved over. All right, in that case, we would have lost 2 phenotypes. Totally random. Can't predict what's going to happen until it already does. A really good example of this is actually in the Amish populations of Pennsylvania. All right, so they're a relatively secluded group of people, and it just so happens that when their population started, all right, there was a founder effect, and that the individuals that started this community happened to have this, I don't want to call it a disease, a mutation, where they have 6 fingers instead of 5. All right, so this mutation is actually pretty common throughout the Amish populations in Pennsylvania. Because they're very secluded, they don't have many other individuals coming in adding more genetic variation. Right? They just happen to have this frequency. All right, so I have a couple questions for you to answer on your own here. And I want you to go through and answer, is this a founder effect? Is this a bottleneck effect? Is this even genetic drift? All right, so give these questions some thought. Work with your neighbor here. All right, have we had a chance to answer all the questions? All right, I'm going to assume that's a yes. I did start handing back your biology of skin color. If you answered all the questions, I gave you 2 points. So you might not see anything on it. That's okay. I gave you 2 points. All right, so let's start with the first question here. All right, we have a population of frogs, and through predation, eventually we only have these light-skinned frogs remaining. All right, is this an example of genetic drift?
No.
Why not?
It's not random.
It is not random, yes. This predator is specifically seeking out those dark-skinned frogs. Right? It is not random. All right, so if we look at the population of northern elephant seals, all right, they were reduced down to 20 individuals. Since then, their population has rebounded to over 30,000. Is this a founder effect or a bottleneck effect? Bottleneck, yes. And then our last one here, we have a group of squirrels. Let's see, they get separated from the rest of the population. They're now confined to a new island and begin a new population. Founder effect or bottleneck? Yes. All right. All right, any questions about genetic drift? All right, so we're going to move on to one of the other requirements for Hardy-Weinberg equilibrium is that there's no migration. In other words, another way of saying that is no gene flow. All right? But we know, again, this is typically not the case. There's typically going to be some gene flow. All right, so gene flow is occurring when individuals migrate between populations that have different allele frequencies. So if we look at the example I have on the screen here, I have a population of western deer and eastern deer. Right? The western deer tend to be more light-furred. The eastern deer tend to be more dark-furred. Right? Now, there happens to be a pass in the mountain range that separates their two populations, and that allows for migration. Right? So we can get some individuals migrating to either side of the mountain range. Now, when this happens, all right, we're tending to reduce the differences in the allele frequencies between the two populations. All right? So originally, we had a lot of light-furred individuals, a lot of dark-furred individuals. Migration has happened, so now we're starting to see some light-furred individuals introducing genetic variation into this dark-furred population, vice versa. All right, so not only does it reduce the difference of allele frequency between the populations, we're enhancing the genetic diversity within each population, all right, on either side of this mountain pass. We're increasing the genetic diversity because we've introduced new alleles. All right, and I wanted to kind of emphasize migration here that we're not just changing one population, right? Both populations are going to change in their allelic frequencies, right? So essentially I have this pie chart representing two different alleles. We took a subsection of these individuals, moved it to the other side, right? It's not only changing the allelic frequencies, right, the number of blue and green individuals here in our one population, but also in our other population, right? One is increasing in green individuals, one is decreasing in green individuals, right? So both populations are going to change when there's migration. All right, I think migration is pretty easy to understand. So we're going to move on to random mating. All right, so Hardy-Weinberg, we're assuming random mating. Mating happens totally random within a population. We're not picking and choosing who we're mating with. All right. Hardy-Weinberg, we know that to be true. In nature, that's rarely the case. All right. It's usually only random if it's difficult to detect some sort of difference between your partner. All right, and this image up here, I believe, is a sea snake. All right, this image was given to me by a colleague, and I tried as I might, I could not find an example of non-random mating, random mating online. All right, it's really hard to find in nature. All right, so I believe this is a sea snake. I'm assuming between different sea snakes, they just can't tell the differences between them. All right, so they're randomly mating with each other. There's no preference associated, because it's difficult to detect. All right, so we rarely see random mating, but we most often see non-random mating. And when we're talking about non-random mating, depending on how we're non-randomly mating is going to depend on what our resulting genotypes might look like. All right, so if we have a sort of mating, all right, individuals with similar phenotypes are going to mate with one another. All right, so we have these beetles here. They are both brown colored. They're attracted to each other in a sort of mating. Once they mate together, they're going to have other individuals that look very similar to them. All right, it tends to decrease heterozygotes. All right, so maybe these are all, let's say, little b, or big B is their genotype. Different than dissociative mating, right? This is where opposites attract, essentially. So dissimilar phenotypes are mating with each other, and we are creating individuals that could be, see, this one's like a darker tan, kind of in between the two, right? Incomplete dominance is happening here, right? Or we might have a mixture of brown and white, all right? But we're increasing heterozygotes, right? We're keeping that allelic diversity there. Now we also have a way that non-random mating can occur, and that's through inbreeding, right? Sometimes a population doesn't have a choice, all right? They have to breed with one another because they might be so small in numbers, all right? So we're mating two individuals that are genetically related. So if they're genetically related, their genotypes are going to be fairly similar, all right? So when inbreeding happens, we're not favoring any particular allele, but we are increasing the likelihood that the genotypes in each generation will be more and more homozygous. That might be homozygous recessive or homozygous dominant. Right? So we're not changing the allele frequencies, but we are changing the genotype frequencies. All right, a good example of an inbreeding example that we see in the United States is these Florida panthers. All right, so inbreeding also has some detrimental consequences. All right, so especially if we have recessive alleles that will pass on a loss of function mutation. All right, so if you have a homozygous recessive individual, some sort of mutation is going to show through that's going to be detrimental to your well-being. All right, normally they're rare. All right, and normally they are masked within heterozygotes, but when we have inbreeding, we're increasing the chance of homozygotes, and this can become more common. All right, so we see this in our Florida panthers, that if we look at the Florida panthers population on the bottom, all right, the percent survivorship of their offspring as they grew up steadily decreased, right? Due to some inbreeding genetics, something was causing survivorship to decrease in their population because they were so inbred. Right, now they actually did a little study where they took a panther or a subset of panthers from Texas, moved them into Florida to increase that genetic diversity. Right, so we had some migration going on, right, and that improved their survivorship. Right, they saw an increase in survivorship because they introduced new genetic diversity. Right, so it's actually a really interesting article on these panthers. This isn't the end of this story here. We're seeing that genetic diversity is starting to decrease once again. I linked the article if you're interested in reading more. All right, so I have a group question for you here. The first one is pretty straightforward. This bottom one, I want you to give me a reasoning why this is random mating or not random mating. Knowing that these are coral polyps, they don't have eyes, they have very little sensory information, all right, hard to pick and choose. All right, so answer these questions together. These should be pretty straightforward.
Yeah.
What do you mean? How do we know? How do we know what? Because Abraham like that's like literally wrong.
Animal behavior questions here. All right, so I think that should have been enough time to answer these questions. I guess they're relatively easy. So which of those mating pairs, all right, the green and the black or the green and the green, represents disassortative mating? One's the same color or a different color? Different, yeah. I like to remember this, the disassortative or assortative, right, because it has the word sort into it. And if we're assorting these by color, right, we're putting them like into the same category. If it's disassociative, we're putting in different. All right, so what about the bottom example? All right, we have these coral polyps. They basically mate by shooting egg and sperm into the water columns in the ocean, having it mixed together to create the zygotes. All right, is that an example of random mating?
Yeah.
How so?
They don't know where they're shooting.
That is exactly right. They don't know whose sperm and egg will end up together. It's totally random, right? And this is one of the few examples I could find online of this being a true random mating event or true random mating organism. All right. If my computer doesn't just freeze up on me, which it might have. I have been signed out. Ooh. All right.
Oh no. It's gotta be done, honestly.
We could take this time to get ahead on your notes if you haven't already.
I'm hashtag down with them.
All right. All right, so let's move on to the next requirement for Hardy-Weinberg equilibrium, which is that there's no natural selection happening. All right. Again, in nature, we know this is hardly the case. There's going to be some natural selection likely occurring. All right, so we've kind of gone over natural selection. All right, we know that's a differential in survival and reproduction. Some individuals are going to survive better and reproduce more than others. All right, and I put this image up here. I know we've gone over this before, just to reiterate those 4 requirements for natural selection. All right, we have to produce more offspring that can survive. There has to be some sort of phenotypic variation with the fitness associated with that variation. And that variation has to be passed down to its offspring. It has to be heritable. All right, so if you had me in lab, you probably saw these images. And if you attended lab, which I'm assuming everyone attended lab that past week before Thanksgiving, you talked about fitness, right? So we talked about relative fitness, which is an individual's contribution to the gene pool relative to another individual's. So if we look at these birds, the one on the top has a lower fitness than the bird on the bottom, simply because it's not producing as many offspring that are reaching reproductive maturity level. All right, so this one's 2, this one has 4, much higher fitness. All right, any questions on fitness? I feel like this is, at this point, you should be pretty familiar with fitness. I'm just going to keep moving on. All right, likely you've heard this example before of the peppered moth. It's a pretty classic example of natural selection. All right, so we have our peppered moth that before the Industrial Revolution, all right, our trees were lighter in color. The light individuals blended in more. Natural selection tended to pick off those that were darker in coloration. All right, now after the Industrial Revolution, we put a lot of pollution into the air. the trees became more or less soot covered, and these peppered moths that were dark and once stood out against the tree now blended in perfectly, right? And now what we're seeing is these light-colored moths are being selected against more often, all right? It's a pretty classic example. I threw up some data just representing that as well. The frequency of dark moths over time, that should be the frequency of light moths. That is a typo there. Frequency of light moths has decreased because after the Industrial Revolution, they're more likely to be picked off. All right, so instead of frequency of dark moths, that's frequency of light moths. All right, big picture of natural selection. The environment is doing the selecting. Right? We're not selecting for genotypes. Right? We're not selecting for the underlying big A, little a, or little a, little a. We're selecting for what is actually physically seen. Right? And there's a lot that can be selecting in the environment. Predators can be selective. We can have disease that might selectively take out individuals, competition, even food resources. Right? So we often talk about natural selection in terms of predation, but know that there's many other factors out there. All right, so natural selection is kind of this blend of chance and sorting. Right? It's kind of chance that one individual's phenotype happens to be better camouflaged in a certain point of time than another, and that they're sorted out. All right? So because of this sorting, only natural selection consistently leads to adaptive evolution. Right? Evolution that is adapting to their environment. Right? So if we think about genetic drift, where all of a sudden we've gotten rid of some alleles through some bottleneck effect, maybe those alleles that were randomly picked away, right, were actually beneficial to those animals. Right? It just happened to be that they got the least beneficial alleles in their population. We don't know. Right? It's totally by chance. Right? Natural selection isn't really by chance. We're acting upon some sort of force. All right, another big picture with natural selection is that we cannot make perfect organisms. All right? Selection can only happen on existing variation. If that variation doesn't exist, we obviously can't act upon it. Evolution is going to be very limited by historical constraints. So whether that might be like history in terms of human civilization with the peppered moths, or maybe it's the history of their genetic diversity. Maybe if a certain individual would be better camouflaged into the grass, for instance, by being green, great, awesome. But if that green allele did not exist, all right, it hasn't evolved yet, and we can't act upon it. We often have compromises, all right? So we generally talk about, oh, this frog is green, and that makes it better for survival, right? But different features can have many, many functions. All right, so if we look at maybe the moths, for example, maybe the white-colored moth, all right, not only helped them camouflage in before the Industrial Revolution, but maybe it also played a role in sexual selection, right? I don't know that for a fact, just giving that out as an example, but we can have multiple functions for a single feature. All right, so all these together, chance, natural selection, environment, they all interact and often very unpredictably. All right, remember, natural selection has no goal. So when we talk about natural selection, we have these patterns that we can start to see in our populations depending on how natural selection is working. All right, we're going to start with directional selection. So this is where individuals at one extreme of some sort of phenotype have a greater reproductive success. All right, so that might be maybe a new allele was introduced into the population that happened to have higher fitness for those individuals. Or maybe there was some sort of prolonged environmental change where the environment that they had originally adapted to has shifted and changed, and now this population is readapting to that new shift. All right, we're going to see, instead of our normal distribution here, all right, that distribution is going to shift one way or another. All right, so your book gives this example of those mice again. All right, so we have these mice. They have white and brown fur. All right, now all of a sudden I should back up. This mice is a population of mostly white fur, right? But some mutation introduced the brown furred individual. All right, they tend to be less susceptible to predation when they're brown because they can camouflage into the forest floor. Over time, they'll be selected for, and we end up with a population that tends to be more brown. All right, and we can see that. Our shift has gone from light-furred individuals down to dark-furred individuals. Right? There is a directional shift in our population. We can also have a stabilizing selection. All right, so this favors the survival of individuals with intermediate phenotypes. All right, so the extreme phenotypes, maybe something's really dark and maybe something's really light. All right, those are going to be selected against, and the ones that are in the middle are going to be favored. A good example of this is clutch size. So if birds have too many eggs, some of those are gonna die from lack of care and food. All right, birds can't tend to all the eggs in their nest. Too few eggs, right? They're really not contributing enough to the next generation, right? So maybe if they only produce 2 birds, both of those birds die, or maybe half of those birds die. All right, not all are going to survive, but if they have just kind of that Goldilocks number, all right. That's exactly what stabilizing selection is looking for. Right? So rather this large drawn out curve, we have this really steep curve where the middle number is favored. All right, the next one we have is disruptive or diversifying selection. And this is kind of where we're taking our nice bell curve here and splitting it into 2. All right, so instead of with stabilizing selection where we favor the middle number, in this case, we are favoring the extremes, and the middle phenotype is kind of being washed away. All right, so likely to occur in populations that occupy heterogeneous environments. So the environments have different extremes to them. And the fitness values are going to be higher for those extreme numbers. So a good example is colonial bent grass. All right, so in this bent grass, we have a population of grasses that some individuals have a very metal-resistant allele to them. They can grow on polluted soils. Other individuals cannot. All right, they're really sensitive to metal. All right, those in between, they don't really do well on either or. They're not really great at growing in a metal polluted environment. They're not great at growing in a pristine environment. All right, so what we see is that population kind of splitting in half. We only get those that are metal sensitive. We only get those that are metal resistant. All right, the last one I wanted to point out to you is this balancing selection. All right, so in this balancing selection, we're maintaining genetic diversity. We're balancing out all of our alleles. We're not shifting one way or another. All right, we're kind of preserving what we have. All right, 2 common ways that this happens is where a heterozygote is favored, and that's called the heterozygote advantage. All right, a really common example, and I'm just going to flip ahead quickly here, is in sickle cell disease. Right? So if you're homozygous recessive, you have the sickle cell. If you're homozygous dominant, you don't develop sickle cell disease, but you tend to have less resistance to malaria. So if you're an individual that has a dominant and a recessive, right, you're not developing sickle cell disease, but you still have resistance to malaria. And we see this, if we look at a map where an area of where malaria is common, and an area where this allele that infers kind of resistance to malaria, you see there's a lot of overlap. Right? So that would be the heterozygote advantage. Another one is a negative frequency dependent selection. Basically, no genotype is going to become too rare or too common. All right? We're kind of balancing out what we have. All right, let's put these into practice. Couple questions for you to answer here. These first 2 should go by pretty quick.
I was reading about the Omicron variant is in the U.S. now.
I believe it.
Okay. Have you done Crystal? No. I'm hoping they'll never get to that point in their life. Okay. I have a very important question. FTS did you take?
But what?
What FTS did you take?
Oh gosh.
This is very important to me.
Scandinavian exploration. Like Scandinavian explorers that traveled the continent.
I thought maybe we took the same one, because I'm in the physics one right now.
Oof, no. All right, it got very quiet, and I'm assuming that means you've answered the questions. So, in this population of birds here, what type of selectional pattern is occurring? What? Directional, yes, directional. We are seeing a shift to the right of a larger body size, right? We are shifting our entire population. It is a directional shift. What about mortality rate in babies? We are stabilizing, yes. The extremes are not favored, right? Kind of the middle ground is favored, right? So babies that have really low birth weight and really high birth weight tend to have higher rates of mortality. All right, I have another set of questions for you to answer. So read each scenario and specify the type of pattern. All right, so instead of looking at a graph, I want you to read a scenario.
Guys, I have a really very important question for you. You want to join me on this? Wow. The biggest anyone else have the issues with gross? The biggest thing you ever want to
I sent you beforehand.
Okay, that was
Not
it.
after.
Okay. But then, I couldn't tell you like 2 years ago.
I seriously don't know.
It was kind of funny to be honest.
Yeah.
Maybe whatever solution it's in. Oh, but this is like 13 pounds. That's not even the 22 pounds.
Contaminated for such a crow. Yeah.
This is so weird. I was like,
I don't know. A pH of 2, that would probably start melting the plastic containers.
I love it. We're going to calibrate. How? I had a pH in like pH was like 2, which is like what is that stomach acid moving inside?
Yeah, it's extreme.
You need people to touch it.
No.
We have like 10 seconds to use some probes before it.
And it didn't work still?
They'd have to C-section.
Well, yeah.
They'd have to C-section.
If you're trying to have a natural birth, like
22-pound baby.
that was the largest baby.
Ever born?
Yeah, 6 pounds.
Oh,
I know. It's like a whole. Were you creepy? Like,
poor mother.
I know.
All right, let's come back together here and answer these questions. All right, so our first scenario, we have birds, sea cracker birds that either have large beaks or small beaks, but very, very few have medium beaks. All right, what type of pattern are we going to be looking at? Disruptive, yeah. It's going to be kind of like a hill with a valley shaped in our graph, right? We're favoring the extremes. All right, so Madagascar hissing cockroaches. All right, we've all had to handle those in our labs. They live in a woodpile and are eaten by lizards. The lizards, they have really small heads, so they're unable to eat the very largest of the adult cockroaches. And instead, they only have prey upon the small and medium ones. So over time, those large-headed lizards are going to survive. That probably should say large-headed or larger cockroaches should survive. In either case, we are favoring one side of our phenotype. What type of selection would occur? Directional, yeah. We are favoring either the cockroaches that are large and cannot be preyed upon the lizards very well, right? Or we are favoring the lizards that have larger heads and can eat more cockroaches. All right, our last scenario here, we're looking at birds. All right, birds in a clutch size. And if there are more than 5, the parents can't adequately feed the young. If there are fewer than 5, the predators may destroy the entire clutch. What type of pattern are we seeing here? Stabilizing, yeah, those extremes are not favored. All right, any questions with these patterns? All right, on the exam, I will probably ask you about the patterns and either give you a scenario or give you a chart and have you interpret it. All right, so I want to point out that your book mentions the grants, All right, so they're a researcher couple, which I believe, gosh, many, many years ago, they actually came to Gustavus for one of the Nobel talks. All right, but they focused their work on one of the islands in the Galapagos, the Daphne Major. All right, so that's our island there. And they looked at the resident finches. Right? And they happened to be looking at these finches during a year that a massive drought occurred. So they had been measuring the beaks of these finches, and they kept measuring it throughout the drought. And the average beak depth tended to increase, right? Because those beaks could basically eat larger and harder seeds. All right, so we kind of saw a data here. So these are the parents. I want you to specifically look at the offspring. Right, the beak depth went from 8.8 millimeters to about 9.8. Right, what type of selection is occurring here? What pattern? Directional. Yeah, we're shifting the mean of the population to one side. Yeah. All right, so I thought this was a kind of cool example that I wanted to bring up. I did ask you to watch the lecture or that video on sexual selection. I did throw up one slide just as kind of a review here. There's a lot of different forms of sexual selection out there. Some of them can be super interesting. All right, but we have intersexual selection, which is kind of like the male-to-male competition. Intersexual selection, which is female's choice. All right, we tend to get very flamboyant displays in one aspect or another. Right, I just listened to a really cool podcast on jumping spiders where they do their dance with their legs. So that's a form of sexual selection. I linked this video to an older Attenborough video. So if you like Attenborough, this will take you there. But there's so many different forms of trying to attract a mate. From gathering up a lot of blue items and piling it together to making these elaborate patterns in the sea floor. To trying to shimmer, all right? Attracting males by shimmering. All right, so kind of the culmination of this lecture here is we can move a population away from the Hardy-Weinberg equilibrium. And that can happen in a variety of ways. Genetic drift, all right, so random chance, natural selection, migration, which is also known as gene flow, and mutations, which we didn't talk about, but we've talked about in the past. All right, so why is the Hardy-Weinberg model useful? Well, it gives us a baseline to begin with, all right? And then when we see us drifting away from that baseline, we can start to examine what might be causing the allelic frequencies to change. What factor is being imposed upon this population? And it could be multiple factors. It doesn't have to be just natural selection or just genetic drift. All right, I'm actually impressed we got through all those slides, but a lot of them were examples. I did put in the additional resources, that genetic drift simulator, if you wanted to play around with it. But that's all I have for you today. Use this time to study for your quiz on Friday.